We call the app, binning, metagenomic binning. And the goal of it is to take environmental samples and reconstruct near-complete genomes from them. So for our purposes today, by an environmental sample, we're going to mean a set of short reads collected from some location. Note that we're talking about DNA, not ribosomal RNA. Although ribosomal RNA has been hugely useful for many tasks, that's not what we're talking about today. We're talking about environmental samples that are taken from DNA reads. Now, the actual samples should be deep enough to support extraction of complete genomes. If they're too small, if the sample's too small and there are too few reads, we will not produce any decent genomes. On the other hand, they should be small enough to allow assembly. Some samples are huge, and assembling them is problematic on our system. So given an environmental sample, here's what we basically assemble the reads in the sample, producing contigs. Alternatively, we'll allow you to supply us with contigs instead of reads. We partition the resulting contigs into bins, and we think of each bin as representing a genome that we're reconstructing. Once we've processed the contigs and placed them into the bins, we keep the bins that contain just one more or less complete and accurate genome. It is important that you can determine whether or not a bin contains a good result. Assuming that we can determine this, we basically construct a bunch of results, many of which are bad, but they're not serious because we don't keep them. We only keep the good results. As long as we can effectively recognize a near-complete reconstructed genome, we can afford to tolerate the bad bins, put them aside for later processing. Okay, let's go to the next one. Why would one bin the contigs in a sample? Well, if you think of the tree of life with your genomes at the leaves, this has been a hugely useful, powerful perspective for studying many of the central issues in microbiology. And so extending this view by adding leaves to the tree that is more reference genomes, one improves it. And one improves it for the same sort of reasons one improves microscopes. It's a fundamental tool. Now, most microbes cannot be cultured, which restricts our ability to acquire complete genomes, which in turn limits efforts to accurately characterize evolutionary relationships. So we're trying to address this issue by extracting new reference genomes directly from the samples. And we believe that extracting complete genomes from metagenomic samples will be the key to acquiring genomes for the unculturable organisms. I've never seen this. Metagenomic samples will play an increasing role as tools for characterizing the state of a patient. Oh, okay. Yes, I'm sorry. There was this fundamental use, and now I'm talking about the use in diagnostics. Besides the fundamental use, one can use reference genomes to create diagnostic tools. The more accurate the set of genomes in a sample that can be reconstructed, the more valuable will be the diagnostic tools we can build. In general, we wish to study microbial communities using metagenomic samples, and we consider the key that limits the quality of such an analysis to be the collection of reference genomes in their quality. So we're creating this growing body of reference genomes. So that binning environmental samples will play a major role in improving our collection of reference genomes, and that's sort of the summary of what I intended to say.
So, this is Bob now. So I want to, with that introduction, say a few words about the service that we brought up. This has been the result of work over the last several months here, and through the development, we've run several thousand samples through the pipeline. And just to give you some ideas of the size limitations we have, our successes anyway, that the largest sample that we've run is about 20 gigabases of reads, which assembled to about half a gigabase of contigs, from which we got 50-some bins, 5 of which were judged as good, and we'll discuss in more detail what a good bin means later. We also we've got the service is up and running, and it's fairly new. We've had, it looks like I just pulled some numbers this morning, about 70 users running nearly 200 samples through. So it's getting there, and people can go ahead and use it. And we encourage you to try it out if you've got data that you're interested in running. Up next, we are going to actually demonstrate the service using a sample from the NIH Human Microbiome Project. And here's the sample number in case you want to follow along at home and read more about this sample. The microbiome project, you may be familiar with. They're collecting and sampling fairly comprehensive set of samples from various locations on the human body. You can see the web address of the site here. On the bottom of the screen, you can see our sample number ending in 683, which is the one we're going to be running. It's about 5 gigabases of reads. Something interesting about this project is that they actually provide they've assembled the samples into contigs that, in case you want to do analysis on them without performing assembly. And they lay out their protocol for how they've gone from the raw reads through, they've done some read cleaning and processing, and then they also have some documents that go through their assembly process. What we're going to show you today is how to submit from both unassembled reads and from reads that have already been assembled. The data files at HMP are also available at the NIH's short read archive. You can see here that the sample that we're looking at actually came from 2 separate runs. And if you read the protocol documents at the HMP site, you can see how they combine those samples, combine the individual runs into a single pair of read files that they then assemble. So we're going to switch now to our live demo at the PATRIC site, and we're going to go through the submission of reads and contigs and look at some results that we've got. So in the PATRIC system is centered at the PATRIC website at patricbrc.org. If you go there, you'll see our page here. There's a ton of information on here. What I'm going to start out by doing is logging in. For any of the computational services at PATRIC, you need to be logged into this site. This lets us, track usage and give you some storage space to work on and the like. If you don't have an account already, you can click on the register button at the top right of the window. This will give you a little registration dialog where you select a username, enter your name and information, and so on. Sort of your regular stuff for internet logins. I already have an account, of course, so I'm going to go ahead and log in. Once you've logged in, you can tell that you're logged in because up at the upper right of the web page, you'll see a little person icon that will tell you your username. So what I'm going to do now is upload my data and start running one of these jobs. To do this, I'm going to use one of the drop-down menus you see at the top of the page. Services is the entry point for the PATRIC computational services. You can see we have a number of them here. I'm going to select metagenome binning. This gives us the input dialog for the binning service. I want to point out I want to point out at the top here, there's a little bookshelf icon. If you click on that, you will get to a tutorial on using the service. A number of our services will have this bookshelf icon that will take you to tutorial information. You can also browse the tutorial website once you get here. We've got tutorials for a number of the services at PATRIC. So the dialog that you see here is similar to many of the service submission dialogues at PATRIC. So I'm going to go through this a little bit because it's a good knowledge to have for using the PATRIC site. The overall flow we're going to do is to upload data files into my workspace. I'll explain what that is in a minute. Provide some parameters for the service execution, then submit the service to be run. The workspace at PATRIC is essentially a distributed file system that we use for storing your input data and your results for the analyses that you run. And you can upload data to your workspace for PATRIC to operate on. What I'm going to do for my first example is to upload a pair of paired-end read files into this upper right segment of the display that tells us the paired-end library. To do this, I'm going to first click on this folder icon next to the read file 1, which will let me define my data for this file. So when I click on that, I get a display that says choose or upload a workspace object. This gives me a view of the current contents of my home directory workspace on PATRIC. You'll see it says rasuser25 patricbrc.org/home. That tells me that I'm in my home folder at PATRIC. What I'm going to do is create a folder to store today's project, so to keep it separated from the rest of the things that you see here. To do this, I'm going to click the Create Folder icon, which is the folder with the plus sign on it. I'm going to give it a name. We're going to call this binning.wednesday. So I've created a new folder. I see it show up here. I'm going to double click. Now I have my new empty folder that I can use to store my data. Next, I need to upload my data files. Remember what I got here by clicking on read file 1. So I'm going to upload my FASTQ data for the first read file. To do that, I will click on this folder or this computer with an arrow icon to upload to the current folder. This gives me an upload file dialog where the upload type is reads, because I'm filling in a reads item in the input dialog. I'm going to give it read files here. I'll click on select file. You can also drag files into this display out of your file browser on your computer. Okay, I'm going to start out with my FASTQ file 1 here. It's named with the sample name that we were discussing earlier. After I select that, I get a listing down here that says the files that I've selected for upload, the type, and their size. I click on Start Upload. If you look at the bottom right of this window, we see we have an upload status bar. This is telling me that I have one file being uploaded that's in progress, and it's currently 24% complete. I can go ahead and continue filling out this form while my files are uploading. What you'll need to keep this window open, keep your browser window open while uploads are going, otherwise the upload process will terminate because it's the browser that's doing the upload for you. So I'm going to load my other read files, same way I loaded the first one. I'm going to switch into my Wednesday folder again. Click the upload button, select file, and this time I'm going to pick my .2 FASTQ file. I verify that I can see. Yes?
We have a quick question here. Is the online workspace connected with a PATRIC command line space? In other words, can you do this through the command line?
You can access the workspace from the command line package. We are going to have command line tools for doing these metagenome binning submissions. They're not there yet, however. That's on the short list of things to add to the command line package. Okay, so I'm going to go ahead and make sure to interrupt me, Ron, if I don't answer something completely for folks. Anyway, I'm going to click on the Start Upload to upload the second file. And again, we see in the status window we've got one in progress, which is the one we just started, one completed, which is the one we started earlier, and we're about 20% complete. Now I'm going to skip the box that says contigs, because right now I'm not uploading contigs. This is an either or. You can select the read library or contigs. And I'm going to look at the right-hand column now, which defines the parameters for the binning process. What I'm going to specify here is an output folder, an output name, and a genome group. The output folder is placed in the workspace where the output will be written. The name is the name that we give to this run so that I can distinguish it from any other runs that I have made on the system. It will also be the name prefix that's used in the workspace for the output for this. So I'm going to fill this in the same way I filled in the other workspace items. I'm going to click on the folder. I'm going to select Binning Wednesday. Remember, what I'm doing is selecting my output folder, so all I need to do is click on that and click OK. So now I've got my Binning Wednesday filled in for the output folder. I'm going to fill it a name. We notice this box is red with a little call out that says name must be provided for the job result. This is telling me that this is a required field for the submission. You see that my submit button is grayed out right now. So I'm going to call this SRS 014683 reads. So this is the output that I got from running the reads on this sample. I'm going to copy and paste this into the genome group name. A genome group in PATRIC is a way that we can store a set of genome IDs for use in further analysis in PATRIC. You can do a number of analyses on sets of genomes. One of the things that the binning service does is generate a set of annotated genomes in PATRIC, and the service will initialize a genome group with a given name containing the IDs of the genomes that have been created by the binning run. I see that my uploads have completed. I got 2 uploads complete. My submit button has lit up, so it means I can click on it, so I can go ahead and submit my job. And we see that I've got a diagnostic that says my job has been queued. If I go down here on the right, I see there's a job status window, which tells me that I've got 1 job queued, 0 running, and a whole bunch of completed. This happens to be the account that we do a lot of our experimental analysis for, so we've run a ton of things through here. So now, what I did by clicking on the jobs window at the bottom is brought up the job status dialog. At the top here, we can see the job we just submitted. Underneath it are all the completed jobs that we've run in the past. We can get a little bit of information about the job, when we submitted it, the service name, the output name, which you'll recognize from the dialog I filled in, the starting time of the job, which was just a few minutes or just a minute ago. It has not completed, and over at the right you can see some details. So this is sort of if you want to check and see what your jobs are up to, this is the place to go to look at it. So I'm going to let that sit and run. I'm going to go and submit our other type of job we're talking about, which is a binning job running from a set of already assembled contigs. To do that, I'm going to again go to the Services menu, click on Metagenome Binning. This time I'm going to leave the read file dialog parts blank. I'm going to fill in contigs. Here I will click on the folder, which brings me to the upload dialog. I'm going to again switch into my Binning Wednesday folder. I'm going to click the upload icon again. Now we see that the upload type is switched to contigs because the field we're filling in is a contigs file. And I'm going to click Select File to pick the file off my computer. I'm going to run this 014683 scaffold FASTA file. I can verify that I've selected the proper file with the right type and verify the size. Go ahead and click Start Upload. There we go. I see my upload is running. The percentage complete is going up. Now I'm going to go ahead and select my output parameters again and show you a shortcut that you can use if you've already selected folders in the system. Notice that the output folder field here is a drop-down. I can click on the arrow for the drop-down and I should be able to get. It's being sluggish. There we go. We get a list of folders that I've been to recently, and I see that this Binning Wednesday folder is up at the top. So I can just click that to select. Now I can select my output name. This time I'm going to call it -contigs because I'm running this analysis from the contigs. And I'm going to copy and paste this into the genome group again, as I did before, and click Submit. Job has been queued. Now I can go click over here onto the Jobs dialog and see that my job hasn't come up yet. I notice at the top left of this it says it's updating the job status list. So now we see we've updated. My contigs run is there and queued for running. We notice that the binning run for my reads is already completed, but there's a couple of new jobs running. The binning process goes through a number of phases. The first phase does assembly and binning, and then for each of the bins it uses the PATRIC PATRIC annotation service to create an annotated genome, and then it does some analysis at the end to generate summary reports. So what you'll see in the jobs display is the binning job running for a while and completing. It may take several hours to run if you give it a large set of reads to assemble. This one ran very quickly because the read set I gave it was a very small extract out of the actual data set. After the binning is done, you'll see a number of annotation jobs running. And then when those complete, you'll be able to see your job results back at the binning, if you look at the binning job. And so with that, I'm going to hand this over to Bruce, who's going to tell you much more about the binning results.
All right. So we have submitted our binning job, and the results will come back in our workspace. Now, a while back, I ran a job just for this webinar so that we'd have the results ready. And here you see I told it that I wanted the output to be called SRS014683.bins. And when you look at this, this is actually a folder, and it's full of files. Some of the files are FASTAs of the bins themselves. Some of them are descriptions of the bins. But the most important one is the report, the thing at the top, which is the binning report. That's an HTML file. And we're going to look at that, and that will tell us what our results were. So there are two tables here. The first one is bins that we consider to be of high quality, and the others are bins of lesser quality. So look at the entire set gives you an idea of what we found, but it's these ones at the top that are the most important. Those are the ones that look like they are, that we have found everything we need to find to make a complete genome. So the very first thing is the Genome ID, and in fact, the bin is stored in PATRIC as a PATRIC genome. And if you click on that ID, you'll go to the page, and you'll see all the standard capabilities and characteristics of a PATRIC genome. We have specialty genes loading here, description of the GC content, the number of coding sequences, and here we are. We see we have 2 transporters, 3 drug targets, two genes that are associated with antibiotic resistance. You can submit this genome to the Proteome service or similar genome finder or variation analysis or anything compared with any other genomes. It is private to you. No one else can see it, but you can use it, but it will show up in all your searches unless you turn off private genomes. They are all called something clonal population, so it's easy to tell which ones come from binning. The species chosen is the species of the closest reference genome that we have in PATRIC. Now, this may not be the correct species. It's usually close. We are working on a taxonomy tool that will give us a better estimate of what the species is. So you've got a genome. We've got its name. The reference genome is the nearby published genome that we derived it from when we did the binning. And then we have several quality numbers. We have two tools we use to determine the genome quality. The second one here is CheckM. This is a standard tool that you can download off the web. It's available everywhere. It gives us two numbers. One is an estimate of completeness. In this case, it says 99.46% complete, which is very good. The other is an estimate of contamination, how much stuff was put in the bin incorrectly. And here it says nothing. The first two numbers are from an internal tool we developed here. And this one uses the annotations of the proteins in the genome. And what we did is we studied the relationships between functional assignments in genomes. And we identified 503 functional roles, we call them, that have predictable relationships. And the tool runs through the genome and says, based on these other roles that occur in this genome, do we expect this one to be here? And if we expect it to be there, and then it's there, we say that's good. If we don't expect it and it's not there, we also say that's good. And for this genome, that happened 92.3% of the time. We then look at the number of times, if it occurs, we look at the number of times it occurs. If we expected it to occur the exact number of times it occurred, then we give it a fine score based on that. Here, 9.2% of the functional roles did not occur the correct number of times. And we say a genome is of good quality, and we spent a long time coming up with these numbers. We say it's of good quality if the fine consistency is 85% or more, if the completeness is 80% or more, and the contamination is less than 10%. And that's what puts them in this top table. We also have some other useful things, the number of contigs, the number of base pairs in the bin. The N50, that says that 50% of the base pairs are in contigs this size or larger. So in this case, that N50 is 84,999. A high N50 means that the genome is probably well assembled. And then mean coverage, if you passed in reads and we assembled them, then, or you're passing in reads from an assembler with the coverage information in the FASTA file, he will pull it out and then show you the average coverage for the contigs in this bin. In this case, 26.65, and that means for each DNA letter in this bin, it appeared in approximately 26 reads. Finally, we have potentially problematic roles. And this is a list of the functional roles that failed the fine consistency test. So our 9.2% of roles that had the wrong numbers turn out to be 48 of them. And if we click on this link, we get a list. And clicking on the link takes you to the results page for the individual bin. And here we have at the top repeated the same numbers we saw in the table on the previous page. And then down below here we have the real meat of it, which is the list of the potentially problematic roles. So we're going to look at some of these and get an idea of what goes wrong in the quality checking and what goes wrong in the binning. The very first one, we predicted that this role should not occur in the genome, but it did. So we're going to click here, and it will take us to the feature display for that functional role in this genome. And I have found that the most useful information comes from the Compare Region Viewer. So what the Compare Region Viewer does is it finds other genes that are very close to the gene of interest. And then it shows the neighborhoods of those genes in the genomes. In this case, well, the compare regions will color red the genes similar to the one that you picked. The square box indicates the region where there was a blast hit. The deepness of the red indicates how close the blast hit was. And then all the genes in the neighborhood are color-coded by protein family. So all of these are the same protein family. All of these are in the same protein family. We have a bunch of genomes of the same genus showing up right here. And you'll notice that they, in general, have the same protein families in the neighborhood of the similar target gene. So what this tells us is that this is a gene that we expect to find in Odorobacter around these others, even though we would not expect that in the average genome. So this tells us that it was the quality checker, not the binner, that had the problem, and also that we made a pretty good guess as to the species in this case. So now we'll move on to a different type of thing that occurs. So I need to find EC27738. Here it is. So here we have something that was predicted to occur once, but it actually occurred twice. So now when we click on the feature display, we're going to see two features in our table. And this takes longer to load because it knows that I don't love it as much as the previous example. So there we go. And so here's a typical feature list display in PATRIC. It's restricted to the two features in our bin that belong to this role that's occurring once too often. And right away you can tell something. Now the local family is a protein family that's restricted to the particular genus, whereas the global family is a protein family that ranges across the whole of PATRIC. So here you can see they're both in global families, but only one of them is in a local family. We suspect that the second one, because it's not in an Odorobacter protein family, doesn't belong in the Odorobacter bin. So now we're going to go to this feature and bring up the Compare Region Viewer. This is the feature that we suspect does not belong. And lo and behold, we see that the protein that's similar to it is in a bacteroids, not in an Odorobacter. Furthermore, we see we're close to the end of the contig. So if we expand this out and update, so now we're going to look at a bigger slice of the contig. You can see this is a short contig. So we suspect this is something, this contig belongs in the bacteroids bin, not the Odorobacter bin, and that's why we have this extra role in there. So we would go out and we'd take the contig out of the FASTA file for this bin, throw it in the other bin, run the two FASTA files through the annotation service again and see if we get better genomes. So speaking of that other bin, we're going to go back and we're going to look at one of the problematic roles in the second bin to see something that is another common thing that occurs. So we're going to go here to the 60 problematic roles, and I'm going to go down to EC 6.3.4.5. And here's another one where one's predicted, but we have two occurrences of the functional role. And they're both in local families. They both look like they belong in this particular species. We know we have a different problem. So we look at the first one. And this time I did a little click hold so I can go directly to compare region without going through the feature first. Now it's going out and it's finding all the proteins similar to the one that we're interested in, so we can see how the neighborhood looks. And here you can see this particular feature is at the end of the contig, and it only matches, the box only matches part of this closest gene, and we have similar colors here indicating a similar neighborhood, but only in one direction. So now we're going to go back to the other one. And here we're going to look at the second one. Sometimes I can get that click hold just right, and sometimes I can't. Manual dexterity is not my strong suit. So here we go. And here you see it. We have a small fraction of the gene, and it's the other side of the neighborhood showing up. So in this case, what we have is an assembly problem. The two parts of the gene should have been put in the same contig. Instead, there was a contig boundary right in the middle. So this gives you an idea of what you can do with the bins produced by the binning service, and also how you can use the problematic roles to see what kind of things happened in the journey from taking the reads and copying them into bins. So with that complete, I will hand it back to Bob.
All right. So, we've now processed your genome for your sample. We've generated bins. We've sort of given you a quick overview of what you can do with the quality reports from the binning service. And what next? There's a number of things you can do within the PATRIC system, sort of depending on what you're interested in with your genome. You can use the proteome comparison tool to, for instance, compare each of your bins to the reference genome that they bin to. That will tell you how complete and how similar or different the proteins in this genome in your bin are. You can use the protein family tools in PATRIC to get an estimate of what the occurrence of protein families in your genome, and you can compare it. Again, compare that to the reference genome. If you have a pathogenic species, you can use the AMR phenotype prediction data to look and see what PATRIC has predicted with respect to AMR phenotypes. You can look at the specialty gene analysis. This is the transporter database, some of the virulence proteins, if that's of interest to you. You can use the pathway analysis tools to explore potential metabolic capabilities of your individual genomes or the groups of the genomes, and so on. And we invite you to go to look at the PATRIC website tutorial page at the URL at the bottom. And we have tutorials on many or all of these services at PATRIC. The tutorials are targeted at reference genomes in PATRIC for the most part. But like Bruce said, the genomes that ran through the service are full-fledged PATRIC genomes that you can use any of these analysis tools to look at. And with that, I think that finishes our prepared materials. If folks have any questions, we'd love to answer them.
So post your questions in the chat window if you would, and we'll do our best to answer them. Are you able to take long reads as input?
The current assembly that we're doing is using the Status Metagenomic assembly, which I believe only accepts short read, card end read files. If you the assembly service at PATRIC is able to assemble long reads. So if you had a long read file, you would be able to assemble it using the PATRIC assembly service and then submit the generated contigs from that assembly to the binning service.
Are eukaryotic reads also binned?
No, because the way the binning service works is that we bin based on reference genomes, and the references we're using are the bacterial and archaeal collections at the PATRIC site.
Yeah, just to clarify that, folks, that PATRIC is dedicated towards bacteria. We do have some other organisms in there, but the focus is bacteria. You'll see some eukaryotes in PATRIC. Their purpose is as hosts for some of the other the bacterial species that we have. Okay. These are floating in pretty quickly. Just to follow up on that one, are there any plans to include eukaryotes in the future? I don't think so, at least not in the context of the current PATRIC project. But I wouldn't say it's out of the question, though. What would you say, Bob?
I think that's correct. Yeah.
Okay. Let's see. Are the reads aligned to a reference or not to be put on different bins?
No, we don't. The process takes first when we assemble the reads into contigs. The contigs are what we're matching to the reference genomes. We don't currently anyway. Don't go back and map the original reads into the references.
Okay. How far can we identify phyla, genus, or species?
Let's see. The current system, the identification is based on the closest bin, the closest reference that the genome, the sample binned into. So it will, you'll get a specific strain, but that's the strain of the closest reference. We can't really, I'm not sure what's being asked, but we don't have a way to do detailed analysis other than picking the closest reference.
We are
Correct.
working on that.
Yeah, we do have tools in the works to do, do a sort of genome-wide estimate of closest phylogeny, but those tools aren't ready yet.
Okay. Can you specify somewhere what exact metaspades assembly command is implemented? Similarly, how is the binning done specifically using the reference genome comparisons?
Let's see. The spades command is actually we're not giving it any special options. We're just giving it the read files and the meta flag to trigger the metagenomic assembly. I think we're using spades 3.11 at this point. The binning algorithm, I don't think we have written up for distribution yet, but I can tell you that the general notion is that we've picked a gene that we found to be a universal one-copy gene across the majority of the PATRIC collection. And we look for instances of that gene from all of our, we search the sample for that gene using it as exemplars all of the genes in PATRIC, that gene from all of the genes in PATRIC, if that makes sense.
All the genomes in PATRIC.
All the genomes in PATRIC, thank you. This gives us an estimate of the best hits in that search, gives us an estimate of the genomes that we think are present in the sample. We then use that set of references to accumulate contigs that match the reference genomes. Okay. And these collections of contigs clustered around the references become the bins that we then do the further analysis on.
Okay. Can you upload bins and have them run checkm and local quality check tools?
Not yet, but one of the very next things coming into PATRIC, we will be adding the quality.
I'm begging. I really want this.
And Bruce really wants this. We are adding to the general genome assembly or genome annotation service in PATRIC. We're going to support the check-M and consistency tests and the generation of the quality reports for every genome that you run through PATRIC.
Right. What binning tool does PATRIC use?
It's one that we developed here internally.
Okay. We work on strains of the same species. How confident is PATRIC on strain level? Not sure if I quite understand that question.
I'm going to assume that means that if you have a sample with a number of very close strains in it, will the binning service be able to separate those strains? Let Ross or Bruce correct me if I'm wrong. I have a feeling that it will not do a good job at separating them, because remember that the way we pick our references is based on differences in the universal gene that we're using to find the bins. If the strains are very close and are in fact different in places other than this universal, we will not in fact get multiple references for that sample.
Right. And essentially 6 months ago we could only get bins on a genus basis. Now we're at a species basis, and as we keep working, we hope someday to get to a strain basis.
Right. And if you guys, if you have a particular sample, particularly in the public archives that is of this form, give us a sample ID, and we can use it, put it in our suite of test data as we work on the service.
Okay. Can we convert PATRIC gene IDs into other IDs? We have an ID mapping service that allows that. You can do it, I think, also from the feature page. You can map to other IDs, and then there's also an ID mapping tool. It wasn't really in the scope of this, but we could put this person to that little tutorial.
Yep.
Okay. Can you explain more about the planned taxonomy tool? For instance, high or low confidence in the named taxon assignment.
Can I take that?
What? Who?
Sorry. Wake up the taxonomy guy.
They're so traumatic, our taxonomy guy blacked out the question. They want to know how fine a distinction you're going to be able to make.
Unfortunately, we don't really have a good answer for that. In the tests that I've done so far, we are usually able to at least get it down to the genus level, often the species level. To some extent, it depends on how much of the genome we have, and we are still working on the tool. I guess the answer is we hope to be able to get at least to the genus and possibly species level. Whether we can get it to the strain level is somewhat problematic in that at the moment our reference collection usually only has 1 or 2 strains of a given species. If I can extend my signature analysis to the entire PATRIC collection, then we may be able to discriminate down to the strain level.
Okay. There's 2 questions I'm going to sort of wrap into one. The binning algorithm sounds similar to the NAFAC-Pollard-Midas algorithm. Do we have a comment on that? And the second thing that's related is, have you done an assessment of your binning tool versus others, such as Midas? The 2 people pointing out Midas.
Have we benchmarked against Midas, I guess is the question.
Yeah.
I think the answer is no.
Yeah. Do we have a copy? Okay.
So at this point, we have not.
Okay.
I will say, however, that we are, in the spirit of the other analysis tools that we have at PATRIC, My view of the PATRIC service is as part of our goal is to have an integration of useful services that are available from the community, and we've had some discussion of providing integrations of other binning tools as well in the binning service. We just have been spending a lot of time on the quality analysis and integration of our tool to the currently. But if there's folks have favorite binning tools that they like, and ideally, if they've got samples that they know bin well with other tools, we'd be very interested in what you think of our binning service, and particularly if there are samples that you can give us or references to samples in the public archives that we can use as test data.
Okay. What can I do with the service, with what the service considers a quote-unquote bad bin? That is, all the same things you demonstrated with the good bins or quasi-genomes. Can I take my stuff from my bad bins and do the subsequent analysis, I believe is the question.
Yep, you can. All of the good bins, both good and bad, are integrated into your private genome set at PATRIC. And so you can go through, in particular, it's interesting to go through the problematic roles on the bad bins, and you can you get a feel for what's going wrong with those bad bins as well.
Okay, there was one callback here to the previous discussion about taxonomy, is that to come in on the taxonomy calling, it's more useful in the person's opinion to have both a name and confidence level in that call opposed to just a name. So they would like to see a confidence level in the taxonomic specification.
Yeah, we should be able to provide some sort of scoring along with it.
Okay. There's always a chance that an assembled genome will be chimeric in nature. Does PATRIC check for chimera in genomes?
The consistency analysis will often recognize the fact that you've got inconsistent functional roles within the same genome.
Right. And I think actually one of Bruce's examples, could be thought of as a result of the chimeric assembly, I'm guessing.
The example where it looks like a Bacteroides contig snuck into the Odorobacter?
Odorobacter. In order to sneak in there, it had to have Odorobacter signatures in that contig.
Yeah.
So.
Okay. So I've gone through all the questions I believe that were in this list. I just went through them in the order they appeared. If someone didn't see their question answered, please repost it or, okay, here's another one. There are only 10 eukaryotic systems available in PATRIC. Are you planning to include plant genomes in PATRIC in the future?
I don't. Yeah, that's sort of out of scope of the current, the current goals of PATRIC, but goals change.
Yeah. So I think one thing to remember, or at least the motivation for us putting in the host genomes that we did were in response to for host response analysis. And usually the host that, since this is funded by NIH, the host they care the most about is human, and so we used human and model organisms for representation of hosts. I don't know that it's, I don't know that it would be something that would preclude us, but we would need to check with the program and see if that's something that would be under the scope of PATRIC.
I guess the issue is the P in PATRIC stands for pathogen, and so we would need eukaryotic pathogens before it would be considered within PATRIC's remit. Isn't that correct?
Well, you've got the host.
Yeah.
It's just a matter of, none of us here makes those decisions.
Yeah, that's above our pay grade.
We'll carry forward that request. I can assure you of that. Let's see. Let me make sure we've got a few more things that have come up. Is it possible to upload coverage info along with the contigs if I've assembled the reads myself?
Yes. So right as it stands, we parse out if you actually I should leave this to Bruce.
Yeah, there are like 3 or 4 different ways we parse out coverage information, and one is if you have the standard FASTA ID that includes the coverage information in it and the length. We'll also look for a coverage equals keyword in the comments of the ID, and I cannot remember now which keyword we look for. But if someone, if someone sends me an email address, I will find it.
Yeah, or alternatively, if you send us a sample of your read file or of your FASTA file with the coverage information, we can make sure that we can pull it out.
Yeah, but there's a little program that goes in and looks at the first couple contigs and figures out how the coverage information is coded in there, if it is.
Okay. So the follow-up to that, you may have covered this, is if so, what difference, if any, will coverage info make to the bidding process itself?
Lower coverage contigs are considered less reliable, and they're, during the early phases of the binning where we're looking for markers that indicate how many bins there are and what species they are, we're only going to look at the higher coverage ones.
There's some coverage cutoffs on the early parts of the algorithm.
Yeah.
Okay. There's also a question or a comment here that we didn't show the contig results.
The contig what?
Results. I'm not sure what that means.
Okay,
In other words, we didn't show which contigs were in each bin.
I'm going to check.
By the way, so if you're interested in the makeup of the bins, I'll flip to Patrick here. Because these are actual PATRIC genomes, in the Sequences tab on a genome is you'll see an enumeration of all of the contigs that make up that genome. So what I've done is clicked on one of our bins here and gone to the Sequences tab. When this fills in, you'll see you can get the distribution of contigs that make up that bin.
Okay.
So here we see that we have 700 contigs in this bin, and you can see the length and GC content and other details of these contigs.
Okay, I'll see if this person, if that answered the question. One of the things I just had a general comment to folks is that we have an online or a help service here, a help desk. It's help@patricbrc.org. You can find it from the Contact Us on our website. And if you send a question like any of these kind of questions here, it gets queued up in our ticketing system, and we'll respond to you. Yes, that was the answer that the person was looking for. So.
Okay, excellent.
All right, and one of the things too here is I've posted a couple times during the webinar that we are recording this. I know we went through a lot of this stuff very quickly, but we'll record it, and in the next couple of days it should be up on the website as a YouTube video, and you can go through there and go through there at your own pace. Got a big thank you with exclamation points that this is a great service, so thanks Bob, Gordon, Ross, and Bruce. Okay, another thanks for the informative webinar. Are there any other questions? We're about to close shop here. Okay, well thanks everybody. Again, if you have any follow-up questions that you didn't get to or you have trouble when you try the service out, just contact us at help@patricbrc.org, and we thank you.